Okay, so let's going back to to slide 10, which is the one that we were on in the last video. If we look at the crosses that Mendel did and just try to pretend that we can't see the big T's and the little t's, because Mendel couldn't. Mendel didn't even use the word gene or allele, or all he knew was that he had true breeding tall parent and a true breeding short parent, and that's all he knew. And he crossed them, and he knew that all the offspring out of about 1,000 were tall. So when he crosses the offspring, how do we end up with a small percentage, 25%, being short? And that's where he had to use, some of his math skills and kind of work backwards. Well, how is the short coming? How do we get short in the offspring in the F2s? Okay, well, the F1s must be carriers of the short allele. So let's just keep this simple, and let's just say that we feel like that both parents in the P generation have 2 genes for each trait, because they all came from 2 different plants that were crossed, at some point in time. They came from a sperm and an egg. So he just reasoned that if the tall parent had 2 big T alleles, which allele just means version of a gene. So 2 big T or 2 tall alleles, and the short parent had 2 short alleles, then the only allele that could come from the tall parent is a tall allele. The only allele that could come from the short parent is a short allele. So the F1 generations must have all inherited a tall and a short allele. They must have this genotype, big T, little t. So they must be tall, but carriers of the short allele, able to pass that one on. And what you're looking at here is a Punnett square. This Punnett square represents 2 of the F1s are the parents. So you put their alleles on the outside of the squares. So you've got 1 parent that donates, and this is the female version. So this is the alleles that were in the eggs. In the eggs, we had big T, little t. And in the sperm, we also had big T, little t, because we're crossing 2 of these F1s. And if you show every possible combination of the alleles of the parents, you can get 2 big T's, which would give you a tall plant. You can get a big T and a little t in 2 different ways. So that combination would give you a tall plant, because the big T apparently is dominant over the little t. But sometimes you get 2 little t's, and that's one fourth of the time. And that's where you get the 25% short. Okay, so that's how all of these traits are working. You have, for every trait he studied, you have a dominant version and a recessive version of the trait. And if you cross 2, what we call hybrid or heterozygous individuals, that would mean they would have one allele from each trait. They would carry the dominant and the recessive allele. If you cross 2 of those individuals, then the F2 generation, you're going to get typically 75% of the offspring are going to have the dominant trait, and then 25% are going to have the recessive trait. And that is what led him to develop the law of segregation. And this is what the law of segregation states. And just remember, segregation and separation, mean about the same thing. Each individual has a pair of factors, which we now call alleles, for each trait. So for every trait, there are 2 factors or 2 alleles for that trait. And those 2 alleles came from the parents, because you got 2 parents. 2 parents means you get 2 alleles for a trait. The alleles will segregate during gamete formation. So when the sperm and the egg form, those alleles separate from each other. And some of the sperm get one of the alleles, and some of the sperm get the other allele. And same thing with the eggs. Each gamete, egg or sperm, contains only one allele from each pair of factors. And then during fertilization, the offspring will get 2 alleles for each trait. One will come from the sperm and one from the egg. Now, some of this stuff, we already know it. We grow up, learning about how this works. But Gregor Mendel was able to prove it through his experiments on the garden pea plant and his careful documentation of the statistics. Okay, so each trait in a pea plant is controlled by 2 alleles. This is how it works. Alleles are alternate forms of a gene. So each trait in the garden pea is controlled by 2 alleles, 2 different versions of the gene, like purple and white flower, or green and yellow garden pea color, that kind of thing, tall and short. Now, the dominant allele is the one that masks the expression of the recessive allele if you have an individual that's got one dominant, one recessive allele, which we call heterozygous. So if you have an individual that's big T, little t, they're going to be tall, okay? And we represent the dominant allele always with a capital letter and the recessive allele with a lowercase letter. Alleles occur on a homologous pair of chromosomes at a particular gene locus, which just means a particular place on the chromosome. Now, when we have 2 alleles that are the same, that's homozygous. So 2 big T's or 2 little t's would be homozygous, and I'll write that. All right, so homozygous would be either 2 big T's or big T, little t. I'm sorry. I'm not going to start over for just that. Little t, little t. Okay, so those would be homozygous. Heterozygous would be big T, little t. And this shows you homologous chromosomes, and look, after replication, they're now duplicated chromosomes. So the red chromosome we'll just say is from the mom or from the egg, and the blue is from the sperm. And the red chromosome has, for this trait up here at the top, is going to be represented by G. The dominant allele is big G, and the recessive allele is little g. Then there's another trait at a different location or locus that's represented with an R. The dominant version is big R, and the recessive is little r. So this individual's genotype is big G, big R, big S, little t, big T, in which we tend to write, no matter which order the letters come in, we tend to write the big letter before the little letter. Okay, so some terminology. Genotype refers to the 2 alleles an individual has for a specific trait. So genotype can be big T, it can be big T, or it can be little t, little t. Oh, I'm sorry. Big T, little t, or little t, little t. Phenotype would be tall or short. The phenotype is the physical appearance of the individual. So these 2 individuals would be tall. And this individual with the little t genotype would be short. Okay, so that's phenotype is the physical appearance, tall or short, and genotype is the actual alleles that the individual has for that trait. Dominant is the allele that if you have a heterozygous individual and they are big T, little t, then the big T is the dominant allele, and this individual will be tall. The short allele will be masked or overpowered by the tall allele. So the dominant allele codes for the protein associated with normal gene function within the cell, in this case, tallness. The recessive allele represents a loss of function. In this case, recessive allele is shortness. And now we looked at a monohybrid cross. Now we're going to look, and that was one trait, a one-trait cross. Now we're going to look at a dihybrid cross, because that is the way that Gregor Mendel developed the law of independent assortment is by studying two traits at one time. Now, this is what the law of independent assortment states. So we're looking at two traits at one time instead of just one trait. So maybe he looked at the tall versus short trait at the same time as he looked at flower color. So height and flower color together. And he learned that the pair of alleles or factors for one trait segregate independently of the factors for other traits. So you get all possible combinations of alleles in the egg and the sperm. So what we're looking at here, this dihybrid cross is crossing, it looks like the height of the plant, which can be tall or short. You can see the key down here. Tall plant or short plant. Big T is tall plant, little t is short plant. And the color of the pod that the garden peas grow in can be big G for green or little g for yellow. So he's crossing two traits at one time, height of the plant and color of the pod. And when he does that, he does it the same way. He still has a P generation. One of the parents is true breeding for the dominant version of the traits, which is tall and green, two big T's, two big G's. And then the other parent is true breeding for the recessive form of the traits, which would be two little t's for short and two little g's for yellow. Okay, so in the F1 generation, all the F1s are heterozygous. Big T, big G, little g. So you can see it better. So all of the F1s are heterozygous, but they're tall and green, tall with green pods, because they're heterozygous and the dominant trait is the one that shows up. But they carry the alleles for the recessive traits, so they're carriers. So the genotype of the F1s is big T, big G, little g. So we're crossing two of them for our dihybrid cross. We're crossing two of these individuals, the F1s, big T, big G, little g. So So if we have two parents and both of them are big T, big G, little g, okay, then the combinations they can give in their egg or their sperm are going to be one of the T's and one of the G's. So we can get big T, big G, and that's this. We can get big T, little g, and that's this. We can get little t, little g, which is this. And we can get little t, big G, which is this. So these are all what we're putting across the top of the columns are the possible allele combinations you can get from one of the parents. So we do the same thing for the parent that carries the sperm. And that's going to be the same combinations because that parent is also big T, big G, little g. So you can get big T, big G, you can get big T, little g, big G, or little t, little g. And then you go through and look at all the possible combinations of the offspring. You get 16 squares here. If the first two combinations with all dominant alleles, you'd get an offspring with big T, big G, big G. And then it just goes on. I'm not going to just repeat all these letters that you see, but what we're going to do is look at the phenotype ratio, and I'll blow this up now so you can see it better. The phenotype ratios are, first of all, we're going to count the number of individuals that have all dominant traits. That means they're tall and they're green. That means what we're looking at is individuals that have at least one big T and at least one big G. We can also look at the pictures. Tall and green. 1, 2, 3, 4, 5, 6, 7, 8, 9. And that's where the 9 comes from. So 9 out of 16 are expected to be tall and green. And then the tall yellow is going to be, there's going to be 3 of them. Tall yellow, 1, 2, 3. So 3 are expected